Hello, anesthesiologists. Welcome once again to the Anesthesia and Perioperative Medicine channel. Today, we're going to dive into a topic that is absolutely essential for pushing our specialty forward, a complete guide to clinical research in perioperative medicine. And we've designed this specifically for anyone who's just getting started on this exciting path. I just love this quote from Paul Dirac. It really gets to the heart of what we're doing here. See, research isn't just about getting a definitive answer and closing the book. It's about asking a question so good that it makes everyone else start asking new questions. It's about that spark that lights up a whole new way of thinking. That's the real adventure. Okay, so here's our game plan for today. We've laid out a 6-part journey that's going to take you all the way from a brilliant idea to a published study. We'll start with the absolute fundamentals. Then we'll get into the architecture of your study, draft the blueprint, navigate all the ethical rules, manage the real-world chaos of running a study, and finally, get your hard work out into the world. It's the whole shebang, A to Z. So let's kick things off with part 1, the key research principles. Honestly, think of this as the concrete foundation of a skyscraper. If you don't get this part right, if this foundation is shaky, it doesn't matter how brilliant the rest of your work is. It's all going to come crashing down. So what are we even talking about when we say clinical research? Well, at its core, it's a systematic investigation. That's the key word. It's not just a hunch you have. It's about creating a structured, rigorous process to turn that hunch into generalizable knowledge. Knowledge that we can apply to all sorts of patients, not just the ones in our study. And let's never forget the ultimate goal. All of this is to generate high-quality evidence that actually helps us improve the lives of our patients. Now, here is the fundamental problem at the heart of everything we do. We can never study every single surgical patient in the world. It's impossible. We have to take a small sample, a bucket of water from the ocean, and hope it represents the whole thing. But the second we do that, we open the door to error. We risk our sample not being a perfect reflection of reality. And these potential errors are the biggest threats to our research. These errors really come in 2 main flavors. First, you've got random error. This is just plain old chance. It's the natural variability you'd expect. It can pull your results one way or the other, but the good news is you can usually reduce its impact by getting a bigger sample size. But then there's systemic error, and we have a scarier name for it, bias. This one is dangerous. It's a flaw baked right into your study design that consistently pushes your results in one specific direction. And you can't fix bias by just enrolling more patients. You have to prevent it from the get-go with a really smart design. So where does this nasty systemic error come from? Well, here are the usual suspects. Confounding is that sneaky third variable that's messing with your results. Selection bias happens when the group you're studying isn't really representative of the larger population. Information bias is just bad data. Maybe your measurement tools are off or people remember things incorrectly. And then there's design error, which is when you ask a question that, even if you answer it perfectly, doesn't really matter in the real world. All right, let's talk about something that can be a real headache for everyone, the p-value. Now, there's the technical definition right there on the screen, but let's break it down. Think of the p-value as a surprise-o-meter. It tells you how surprising your results are, assuming your big idea is wrong and there's actually no effect. That's the null hypothesis. A tiny p-value just means your data would be super surprising if there were no real effect, which suggests maybe there is a real effect. But, and this is a huge but, it is not the probability that your hypothesis is true. Which brings us to this classic question. We've all been there. You're reading a paper, you see p less than 0.05, and your brain lights up and says, it's significant, it works. But what does that number actually tell us? Getting this right is absolutely crucial because getting it wrong can lead us to make bad decisions for our patients. It's a big reason why we're moving towards focusing more on things like confidence intervals, which give us a much richer picture. So here it is, plain and simple. A p-value of 0.05 means that if your new drug had zero effect whatsoever, if the null hypothesis was totally true, you would still expect to see results as strong as yours or even stronger about 5% of the time just by pure random chance. That's all it is. It's a check on how compatible your data is with the no-effect world. It tells you nothing about how big or important that effect might be. Okay, we've got our core principles down. Now it's time to become architects. In part 2, we're talking about study design. This is the blueprint you'll use to actually answer your research question. The design you choose is one of the most important decisions you'll make because it dictates everything else that happens. When you really boil it down, study designs fall into 3 main categories. First, there are observational studies, where we're basically just watching what happens in the world without interfering. Then you have experimental studies, where we actually roll up our sleeves and assign an intervention. And finally, you have analyses of published research, where we pull together all the existing evidence to see the bigger picture. Let's start with observational studies. In this case, the researcher is a passive observer. We're just looking at what happens when clinical care takes its natural course. Now, the most important thing to remember here, and I can't stress this enough, is that these studies are great for finding associations. They can show you that two things seem to be linked, but they have a really hard time proving that one thing caused the other because of all those potential confounders we talked about. I think the easiest way to understand these is to think about the direction of time. In a case control study, you're a detective. You find the people who already have the outcome, the cases, and you look backwards in time to hunt for clues about their exposure. For a prospective cohort, you're looking into the future. You find a group with a certain exposure today and follow them forward to see what happens. And a retrospective cohort is like being a historian. You're doing the same thing as a prospective cohort, but you're doing it all in the past, using old records. This table is a fantastic little cheat sheet. A cross-sectional study is just a snapshot in one moment in time. It's fast, but it makes it almost impossible to know what came first, the chicken or the egg. Case control studies are super efficient for rare diseases, but you have to be so careful about picking the right controls and dealing with people's fuzzy memories. And cohort studies are the heavyweights. They're powerful for seeing how things unfold over time, but man, they can be slow and really expensive to run. Okay, so enough just watching from the sidelines. Let's get into the action with experimental studies. This is where we, the investigators, stop observing and start doing. We actively give a new drug or try a new procedure on one group and compare them to a control group. That act of intervening is what gives these studies their incredible power to get us closer to proving cause and effect. And even within experimental studies, we've got a whole toolbox of different designs. The one we all think of is the superiority trial. Is my new thing better than the old thing? But sometimes we ask different questions. An equivalence trial asks, is this new generic just as good as the expensive brand name? A non-inferiority trial asks, is this new, safer drug at least not worse than the current standard? Then we have really clever, efficient designs like factorial studies that let us test two things at once, or cluster trials where we randomize whole hospitals, which is perfect for big picture quality improvement projects. Now, this is a really cool and clever design that's getting more popular, the stepped wedge. Just look at the visual here. Every single hospital or cluster starts out doing the usual care. Then, one by one, at random times, they switch over to the new intervention. By the end of the study, every single site has gotten the new treatment. This is great for a few reasons. Ethically, nobody's left out, and logistically, it lets you roll out a new, complex change in a manageable way. And finally, when we're trying to combine all the existing studies in a meta-analysis, we have to watch out for something called publication bias. That's the problem where exciting positive studies are much more likely to get published than boring negative ones. This funnel plot is our detective tool. Each dot is a study. If everything is on the level, it should look like a nice symmetrical funnel. But if you see a big chunk missing, like we do here on the bottom left, that's a huge red flag that small, negative studies might be missing from the picture, which could make the treatment look better than it really is. All right, you've got your principles, you've picked your design. Now comes what might be the single most important part of the entire process, building your study protocol. This is so much more than just a document. It is the detailed, step-by-step recipe for your entire research project. A great idea without a great protocol is just, well, it's just an idea. The protocol really is the entire life cycle of your research, just like you see in this diagram. It's what gets reviewed by the ethics board, it's the instruction manual for your whole team during the study, and it's the foundation you use to write your final paper. It is the ultimate source of truth for everything you do. So what needs to be in there? These 6 things are non-negotiable. You need a crystal clear hypothesis, super specific criteria for who's in and who's out, a detailed description of your interventions and your predefined outcomes. You have to show your math for your sample size. But I want to linger on number 6 for a second, the data analysis plan. You must decide exactly how you're going to analyze your data before you collect a single data point. This is what keeps us honest and prevents us from just fishing for a positive result. Okay, your protocol's written, it's beautiful. Now you have to get approval to actually do the study. This brings us to the world of ethics and regulations. And please, don't think of this as just a bunch of hoops to jump through. This is the system that protects our patients and makes sure our research is trustworthy. You're going to encounter four main checkpoints. First, you need ethics approval from an IRB, or Institutional Review Board. Their only job is to protect the participants. Second, you have to register your trial publicly before you enroll your first patient. This promotes transparency and stops people from hiding negative results. Third, if you're using an unapproved drug or device, you'll need a green light from regulatory bodies like the FDA. And finally, you'll need a plan for how you're going to share your data with other scientists, which is becoming more and more important.
Now for the fun part. You've got your approval, you're ready to go. Part 5 is all about study management, the nitty-gritty, day-to-day work of actually making the research happen. This is where great plan meets reality, and it requires a whole different set of skills.
To pull this off, you need to be a master of these 5 areas. You're not just a scientist anymore. You're also a project manager. You have to manage the money, the data, and the people. You need an ironclad system for reporting adverse events to keep patients safe. And you need to constantly monitor your own study to make sure you're sticking to the protocol and the data is clean. It's a huge job, but it's absolutely vital. Speaking of safety, this table is so important. The language here is incredibly precise for a reason. An adverse event is literally anything bad that happens to a participant, even if it has nothing to do with your study. A suspected adverse reaction is when you think, there's a chance our study drug caused this. And a serious adverse event has a very specific high-stakes definition. Knowing the difference isn't just semantics, it's a critical part of running an ethical and compliant study. All right, we are on the home stretch. You have done it. You've run the study, you've collected the data, you've analyzed it, but your job is not done. In fact, the most important part is still ahead, sharing what you've learned with the world. And there are right ways and wrong ways to do this. First, don't reinvent the wheel. Use reporting guidelines like the Consort or Strobe checklists. They're amazing tools that make sure you don't leave out any crucial information. Second, figure out authorship early and base it on real contributions, not just who's the boss. Third, be smart about where you publish. And please, watch out for those sketchy, predatory journals. And finally, above all else, maintain absolute research integrity. Be honest, be transparent, and be proud of your work. As we close out, I want you to think of research not as a straight line with a finish line at the end. It's a circle. It's a continuous cycle. You finish a project, and then you have to take the time to reflect on it. You listen to feedback from your peers and reviewers, and you use all of that to plan your next project even better. So I'll leave you with this final thought, a question to take with you. As you start thinking about your own research projects, how are you going to build this cycle into your work? How can you make every single project, whether it succeeds or fails, a stepping stone that makes you a better scientist and helps move our entire field forward? Thank you so much for joining us.